The human body is inhabited by millions of microbes, which are collectively known as microbiota. The gut microbiota refers to the community of microorganisms which inhabits the gastrointestinal tract of humans as well as other animals. These microorganisms coexist in a symbiotic relationship with the host organism. The gut microbiota resides in the colon, which is the last part of the digestive system. Due to the advancement in the DNA sequencing, we have studied these gut microbiota extensively. From 1840s, the research has been going on about the gut microbiota, and the pivotal work of Theodore Estrich, Henry Tessier, and Leah Metchnikoff and Alfred Nissel has advanced the scientific knowledge and applications of these gut microbiota. The Human Microbiome Project, which was launched in 2008 by NIH in the United States, has given us a lot of knowledge by using advanced DNA sequencing techniques to gain a lot of knowledge and advancements in the field of microbiota.
So, gut microbacteria actually plays an important role in human health, such as supplying those essential nutrients synthesis to vitamin K. Also, it also by aiding the digestion of cellulose and also promoting the angiogenesis and entire nerve functions. The gut microbiota is a predominantly composed of bacteria which inhabit the human gastrointestinal tracts in vast numbers, playing essential roles in digestion, nutrients synthesis, immune system regulation, and also protection against the pathogens. These bacteria perform dietary fiber, producing short-chain fatty acid, and contribute to the synthesis of essential vitamins while also modulating the host immune response. This composition of the gut microbiota can vary among individuals and also can influenced by numerous factors, making a balanced and diverse gut microbiota for crucial overall health.
Let us talk about fungi. The discovery that fungi can trigger significant psychological process has recently rekindled interest in research, which has been overlooked for many years. It has been demonstrated that fungal components of the gut microbiota can affect the host immune reaction by reducing or enhancing local inflammatory responses. The most common fungus detected is the Candida albicans, which is accepted as a true gut symbiotic fungus. So, what are the functions of Candida albicans in our gut? Well, a recent discovery was that Candida albicans is a primary inducer of T helper 17 cells, which are a type of white blood cell called lymphocytes that coordinate protectively immunity in humans. Thus, the fungi is a key regulator of human Th17 responses both in healthy conditions and during intestinal inflammation. Next is virus, a key component of the human microbiome. The human microbiome is relatively newly discovered. There are two types of virome, which are eukaryotic viruses that replicate in human cells and prokaryotic, which is ruled by bacteriophage, which is the virus that infect bacteria. In contrast to bacteriophage, which appears to be strongest early during development and thereafter decreases, the eukaryotic virome appears to be gained gradually with age. Although further research is needed, it is proposed that resident viruses play an active role in controlling intestinal inflammatory as they promote intestinal homeostasis.
Gut microbiota also has several benefit effects on human health, including promoting innate and also adapting to immunities, maintaining the intestinal ability internally, helping the metabolism and also synthesis the essential nutrients such as vitamin, carbohydrates, and also resisting the pathogens evasion. But also, butane rate is the main source of energy for human chronologies and can also be induce the apoptosis of these colon cancer cells. And this can activate the intensification of gluconeogenesis, having the beneficial effects on glucose and also energy homeostasis. So butyrate is essential for appeasing cells to actually consume large amount of oxygen through the beta oxidation, generating a state of hypoxia that maintains oxygen balance in the gut. This will prevent the gut microbiota from dysbiosis. As for gut microbiota, also has its importance. So it actually its importance is actually linked to this common health issue like type 2 diabetes and obesity. So it's not just about extracting this because of more energy than those from the food. The microbiome affects factors such as endothelial and gut membrane, insulin resistance, and also those hormone gene expression and interaction with bile acids. Strategies like this, we're actually using prebiotics and probiotics, antibiotics, or even gut microbiome transplant are being explored to manage the metabolism disease like obesity and insulin resistance through the diets. Normally, the function of the gut microbiota includes the host to be as the absorb of these nutrients metabolism, xenobiotic, and so the drug metabolism and maintenance of the structural integrity of the gut muscular barrier, and also immunization and also the protection against pathogen. Gut's microbiota also helps to break down the complex carbohydrates, fiber, and other industrial substance in our diet that also all our own enzyme cannot process it. This fermentation process produce short-chain fatty acid and also other metabolites which can be also absorbed and also used for energy by the host.
Let's look into human diseases related to gut microbiota. The first is obesity. Obesity changes in gut microbiota composition may play a vital role in the development of obesity. Several gut microbiota species, which are called the obesogenic gut microbiota, can significantly contribute to obesity, such as Firmicutes, Bacteroides, Rhizobium, Lactobacillus, and Clostridium. In particular, obesogenic gut microbiota could facilitate obesity by producing short-chain fatty acids such as butyrate, providing the host with an extra energy and inducing low-grade inflammation caused by intestinal microbiota metabolites. Now let us look into hypertension. The ratio of bacteroides and firmicutes within intestinal microbiota has been significantly associated with hypertension. In addition, levels of lactose, which is a byproduct of intestinal microbiota fermentation, are higher in patients with newly diagnosed hypertension compared to healthy controls. Next is cardiovascular disease. Intestinal microbiota has been studied as a contributing factor to heart disease and stroke. Patients with coronary artery disease have altered population of the most prevalent bacterial species that make up the gut microbiota, with a decrease in bacteroides and an increase in firmicutes. Moreover, tourmaline and oxide, a metabolite derived from the gut microbiota, performs in developing atherosclerosis cardiovascular disease. Now let us look into how diet affects gut microbiota. The first is dietary fiber. The adequate intake of dietary fiber helps promote the growth of beneficial bacteria and maintain intestinal health. Saturated fat and cholesterol. A diet high in saturated fat and cholesterol may have adverse effects on intestinal flora. Protein sources. Animal protein and plant protein sources have different effects on intestinal flora, and plant protein sources are generally beneficial. Second is diversity. A diverse food intake helps maintain a diverse intestinal flora. Fermented foods. Fermented foods containing probiotics help increase the number of beneficial bacteria. Antibiotics. Overuse of antibiotics may interfere with the balance of intestinal flora. Last but not least is herbs and plant compounds. Some plant compounds can have positive impact on gut flora.
In this topic, our main focus was to understand what gut microbiota is and how it functions. We found out that these gut microbiota have several beneficial effects on human health, like promoting innate and adaptive immunity, maintaining intestinal health, synthesis of essential nutrients like vitamins and carbohydrates, and resisting pathogens. This topic also sheds light on how these microbiota impact several functions in host nutrient metabolism and immunomodulation. We also learned how these microbiota affects our diet and also causes change in appetite. With the upcoming developments in genetic technology, transgenesis of these microbiota can be done to fight and prevent many diseases. We realize how important these microbiota are in maintaining human health. This topic also focuses on how to improve and maintain the total microbiota health. Overall, this topic has made us learn a lot about the microbes present in the gut and made us realize how important it is to understand and maintain them.